Why did you choose to enter the field of microbiology?
It was because when I did my undergraduate, I really liked the microbiology classes. A lot of people didn't like it because it was smelly. They have all these petri dishes that are incubating, and you would enter the classroom and it would just smell like, I thought it smelled like, I don't know, like chicken broth or so. I wasn't too grossed out by it, but a lot of people were like, "Oh, this is microbes, bacteria," and I'm like, "No, this is fantastic." And we had just had a fantastic teacher. The classes that he taught, like the where you sit with 200 people and listen. He was not in the practical part, but I just enjoyed that. And I think a lot of choices we make, which profession are we going to take, I feel are based on good teachers we had, because you just like the subject more if you like the teacher. And I might have chosen something very different, but yeah, this is what happened. And I'm later my PhD advisor also, so that was great.
Yeah, that's awesome. My colleagues wanted me to ask you the next question. What's your favorite microbe?
There's so many. I don't know. I mean, I've worked on Vibrio cholera, and it's not my favorite in terms of what it can do. It causes cholera. That's what I did my PhD thesis on. But I feel the disease it causes is so devastating and deadly, and it's just amazing. I feel so. It's not really my favorite in that I like it. It's more I'm just in awe for what it can do. I'm just it's just this tiny bacterium that secretes this toxin, and then you get, it's a bit gross, but you get massive diarrhea. You can die of it, and it's just amazing what it can do. And it still causes huge outbreaks all over in the world, and I just am still sort of following what it does and a little bit of research. It turns out it has 2 chromosomes, which I never knew when I did my PhD work on it. So I guess the topic you do your PhD on, that stays close to your heart. So
Who would you say were your greatest mentors along the path to where you are now?
so yeah, I would say I had 2 PhD advisors. One was my daily one, and then the microbiology professor was the other one. So they're both professors now. So I think there have been, I mean, we had difficult discussions, and I think at the time I didn't realize it, but my closest PhD advisor is a very good friend now, not the microbiology professor initially, but the other one. Good friend of me now, and his name is Fritz Moy, but he learned me to really do careful research, like to really, how do you say, dig deep and write carefully and be very thorough. But I also had in the lab, I mean, a lot of people mentor each other. So I had, when I actually first started, before I did my PhD, I worked a year in that same lab. And there I had a mentor who was only a couple of years older than I was, and doing his PhD, and I was helping him sort of as a research assistant. And he was super enthusiastic. And he's like, "Oh, it didn't matter that we were working late." He was just, "Oh, okay, let's do this, Elise." And yay, we have results. I don't know. He was on fire, I guess, and he lighted this fire of being curious and of being, you know, loving your work and piecing together different pieces. I don't know. He was just really great, and yeah, so he inspired me to stay in that lab and do my PhD. Also worked at a startup company, and so I had my supervisor there, my manager, was a woman with a lot of experience in startups. No, I shouldn't say startups. She worked at like Genentech, like big companies, and she taught me a lot about the switch from research from academia to biotech and what to do. And she left at some point the company because, as I heard later, she was suspicious about what was going on there and behind the scenes in the leadership. And yeah, then I also found out, and I left the company, and later the company got raided by the FBI, and so there was a big stink about it, and the founders are being charged and they're fugitives for the US government. So it's a whole story. But she was amazing in that she taught me a lot about how to balance, as a scientist, you want to know and you want to disclose what you do, but as a as a company, you cannot all disclose your secret sources, and so how do you balance that but still do honest science? And so she was just an amazing mentor, and I'm not an easy person to supervise or manage because I'm so convinced that I'm right at some point. And some things she was able to handle me.
In a 2020 Nature article, you were called a super spotter, as you're known for being able to detect the presence or lack of science integrity in published papers. What motivated you to delve into this field?
So it's a long story, and I've told it many times, but I'll try to be short. So the super spotter refers actually not just to spot lack of science integrity, it's to spot duplications in images, which is what I do. I look at photos and compare them. I started doing plagiarism because somebody said, I read about plagiarism or I heard about it, and I just grabbed the sentence that I had written in a review paper, put it in Google Scholar between quotes, and found that somebody had plagiarized that sentence that I had written. That made me very angry, and I'm still running on that anger. Later, I switched from detecting plagiarized text to images because also by accident I discovered a duplicated image in a science article, and it's the same photo, but it was turned upside down, and I recognized it. Then I realized I have this, I guess, super spotter talent, which I think about half of the people have. And I just decided to make this into my hobby and scan papers to look for these duplications. But yeah, I think the initial anger of that plagiarized article where people had stolen my sentence, that just made me angry because, as scientists, we spent a lot of time carefully writing and doing research. And then if somebody just steals your results or runs away with it, just my sense of justice was not very happy with that. And I still feel that anger that I felt on that day. So that's fueling me.
Based on all your experience working in this, do you have any tips for ways that any member of the scientific community or even the non-scientific community could become more adept at recognizing the lack of scientific integrity in published research?
Yeah, I mean, it's hard to know. So I recognize particular problems, and I feel those are easy for me to recognize, I guess also because I have experience and I'm specifically looking out for them. But just reading an article, again, with the idea of could this be fraud, a little bit more suspicious, like we scan our emails, like, is this a scam or not? And I think just being aware that fake papers exist, paper mills exist, and fake authors exist, and things like that, makes you more less naive, I guess, and being able to recognize it. So I focus on images, but you can look at many different things like ethical. Is there an ethical approval? Is there, if there's animal experiments, are the tumors very big? Maybe, is that ethical or not? Is that humane? Is there, if people write that they do the experiments in nude rats or nude mice, and you see a photo of a mice with hair, that's weird. That's unexpected. I've seen all these things. I've seen papers with prostate cancer in which half of the patients were women, and it's very unexpected. And it's things like that. And these are obviously extreme cases. But I think if you just read the paper from the different perspective of it could be fraud, perhaps, then you might be more able to recognize it.
In what ways does your degree and experience working in science inform your perspective and your work in science integrity?
So I did my PhD in microbiology, and not most of the papers in microbiology nowadays do not have images, do not have photos. They have figures of line graphs and ordination plots and things like that. But those are actually quite easy to fake if you really want it. But there's nothing visible that could lead you to wonder if it might have been falsified or fabricated. But I also, within microbiology, I have a background of molecular biology, which are more the techniques like Western blots, gels, sequencing, and so I have a lot of molecular biology background, even though I didn't do a lot of Western blots. I know how they're, I've done a couple in my undergraduate, and I've done a couple during my PhD, not too many, but I have some general idea on how these things work and how they should look like. And so that helps me recognizing things that are wrong. But also in general, I feel as a scientist, you get taught in how to design a good experiment and how to recognize a flawed experiment. If you do an experiment where, let's say you have some magic cure for COVID-19 and you give 5 patients who are sick already for a week and you give them your magic drug, and another week later, they're all better. Like, that experiment doesn't necessarily prove that your drug is working. You know, they might have gotten better if they ate chocolate pudding, which is usually what I say. But like, you have to have a control group, and you have to, you know, cannot just do one group and then say, oh, my thing works. And I think a lot of people who are not scientists make that error, even though it seems very logic to me that you have to have a control group, and it's not always possible, obviously. But those are, you know, poorly designed studies and methodology. Or if one group was much sicker than the other, you know, and you count the number of deaths between the control group and the treatment group, but your treatment group was less sick to start with, that is also, or had more, you know, younger people, the average age. I've seen that. Like, those are maybe less obvious problems, but they're still very recognizable. So my background just as a scientist gives me some of the tools that I need to look at methodology of papers and see flaws in it.
What do you feel being a scientist has taught you?
To be curious, to be honest, and to be diligent in how you report the results and cite others, like give other credits. So I feel those are some general things that I feel maybe they, you know, I already had that in me, but I feel those are things. So I feel for me, science is about reporting the truth. You're curious in how things work. So you do some experiments, you know, get some results. Hopefully they tell you something, they're conclusive. And so you've discovered something. And even though it might be a tiny part in science, as scientists, we also build on each other's work. So we report our findings and we add to the growing body of knowledge of science. And but you also have to give credit to the people before you on whose work you base your work. So I feel those are essential traits that also could transfer to real life. Like, if a person helped you, you thank them, or you give them credit. And if if you've done something wrong, you apologize. You don't lie. You stick to the facts. So those are, I'm not sure if it science taught me that, or I was attracted to science because maybe these are values I already had before I became a scientist. But it definitely appeals to me. And so everything that is misconduct is not science, because if you cheat in science, you're not doing science. That's sort of my philosophy and why I'm doing this.
What advice do you have for scientists who are just starting their career in science?
Find a good lab to do your research in, to work, especially if you're starting your PhD. Of course, in the US, we already have a fantastic system of rotation. So you'll have 2 or 3 labs that you rotate through, and you can pick your lab. But ask around. Don't just interview with the professor. Also interview with a couple of grad students and perhaps postdocs who work there. Sorry. So you want to have a lab where the supervisor, the professor, is around a lot. I worked in a lab. I was already a experienced postdoc, my second postdoc. And so I was experienced, and I did not need a lot of daily supervision. But our PhD advisor, of the whole group, the professor, was always on the road. It seemed like he was occasionally 1 day a week. He was in the lab, but then everybody, he only was there for a couple of hours, and then everybody wanted to ask him questions, and he was just not very available for the young researchers. And I felt sorry for them. And we, as more senior people, tried to help them and give them advice, but sometimes we would our advice would be different from the advice they finally got from the professors, which is confusing. But as scientists, we all have our own opinions, so we might sometimes differ. But yeah, so search for a lab that has a a good leader, somebody who's there, somebody who's actually mentoring, not just only come to me, when you have a paper ready. You want a little bit more supervision and mentoring at the start of your career. And also a person who's not a bully, because if you have a bully or a very demanding person, of course, as scientists, we need to be demanding, and you need to you cannot always stick to the 9 to 5 rule. You probably need to put in a couple more hours. But I've also seen people labs where scientists the researchers were expected to come in also on Saturday and Sunday, and I feel that's just not a good balance. You need a work-life balance, and occasionally I've come in the weekend because your cells didn't grow, or the machine you wanted was finally available, and so you just do it then. But I do feel if the person who is supposed to be your mentor is more worried about the hours that you make than the quality of your results, that I feel it's the wrong lab for you. And so you need to interview people and make sure that you find the good balance of a person who is able to lead a lab and get good results and get good grants and all that stuff. But don't stare blind on the lab that has 10 Nature papers, because that's just the output, and maybe that means that the professor is a bully or very demanding, and maybe just not even mentoring. And if the lab is too big, also I feel if a lab is more than 20 people, or especially if most of them are graduate students, there's just not a lot of attention for everybody.
What qualities do you believe make a good mentor in science?
I mean, there's many things you need to learn when you start to be a scientist. So I mean, you want your professor to be around and to mentor you in terms of the research questions you're going to tackle, and in terms of connecting you to people who can help you with the more practical things. And sometimes mentors might just be another graduate student who is a little bit longer in the lab and knows how to operate the machine that you need or so, and will help you. And I've seen labs where people are super helpful, and I've also seen labs where people are not just the PI, but also the other people working in the lab themselves are very protective, and they're like, these are my buffers, and I'm not going to tell you how I made them, or I'm not going to, I'm just going to point, oh, it's there, but you're not going to get a lot of help from other people. I've tried when I was a little bit more senior in that lab where the PI was almost never there, I've tried to be helpful for incoming people, like the first week, just take them out for lunch, show them where the canteen is, stuff like that, and say, this is how that machine works, or I'll give you some of my buffers, and so you can get your experiment starting, or just tag along with me, and I'll just show you how I do a DNA extraction, and so you learn where everything in the lab is in that first week. And I feel you need to find not just a professor, but also some other people in the lab who can help you and who are willing to share some of their time with you, but also then, be grateful for that. I feel some people did not really, they thought they took it for granted, I guess, but so you need to have people who help you learning how to do science, how to, deal with the troubleshooting. They need to be people who give you space when things don't, hopefully finally they will work, but in the beginning experiments might not work. So how do they give you space to repeat experiments, or do they keep on pushing you for results? Because those people who push for results might lead to misconduct, right? At some point you want to get out of the lab and you just fabricate something to please the professor. And that is, of course, not the type of mentor you want. So people who give, not just, and also people who manage you, not because there's a difference between mentoring and managing. You also need to be taught how to work in a lab environment. And if there's conflicts between people, which eventually there are, you need a professor who can actually also deal with that.
Tell us more about your work with scientific integrity and what specifically you focus on.
So I specifically focus on photos. So I look for duplications and signs of tampering of photos. And so I'm looking for, so the first duplication that I might find is just a direct duplication where the same photo is used twice to represent two different experiments. The exact same photo. The second type of duplication is where two photos overlap. So you can see, they have, a couple of cells in common or a couple of lanes of a gel or so. And the third one is where within a photo, you see elements of duplication. So you see parts of a tissue being stamped, maybe to hide something, or maybe to make it look like there's more of a specific feature or cell or coloring in that photo. And then sometimes photos can be rotated, stretched, mirrored, things like that. So those are very, those might be a sign of deliberately trying to mislead a person. While two identical photos, occasionally that could just be an honest error. If you do a lot of Western blots, a lot of these Western blots look very similar. They're basically, gray backgrounds with black stripes. And so somebody might have grabbed the wrong photo. But if it happens over and over again, that might be a sign for misconduct. So I try to look for these things. I've seen other problems obviously as well, but that's my specific focus. And I am using nowadays software to help me find these things. So the software I'm using is ImageTwin, and it also has a library of images that have been previously published. And occasionally it will find something amazing, like these people have just stolen a graph or a photo from some other, random publication from 10 years ago, and they just stole that and passed it off as their own. That's just amazing. But yeah, so that's sort of my focus. And I have around 7,000 papers that I've reported, mostly online on Papier, but I also around 2,000 that I've reported to the journals. I'm very much behind. And I also give talks about this work, and I'm in a lot of meetings where we deal with, how do you deal with retractions, and that papers are still being cited even though they have been retracted. And so I'm very active on Twitter, showing also the lack of response from some of the journals about these things. And I have a fairly large following on Twitter, so I'm tempted to go to Blue Sky, but because I don't like Twitter anymore. But it's hard to build up a new social media audience on a different platform. So those are some of my focuses.
How do you go about building? Oh, how did you go about building a following in Twitter and then now at Blue Sky?
I guess the Twitter took me years. So I was first just a microbiologist tweeting about microbiome papers, which is sort of that's also my handle, Microbiome Digest. And I think I built up a smallish audience of around 2,000 people within maybe a couple of years. But then when I started to tweet about science integrity, and I announced that I was going to take a year off of my work and do this unpaid and see how it works. And it works out. I can actually make some of a living with Patreon donations and giving talks and being reimbursed for that. I build up. You sort of see in my Twitter. I should do it the other way around. Well, but anyway, it's my Twitter following started to build up even more. Like I was clearly tapping not just into the microbiologist anymore, but also in other scientists' interest. And so I think a lot of people like my challenges. So I do something that people look forward to. I post a challenge. Like, here's a photo, what is wrong with it? And I think people love that, and they get an emoji award. So you have to sort of know how to engage with your audience. And you have to be able to say something that people think, wait, yeah, that's actually really good. Like, so I'm a little bit controversial because I have called out science editors of, big name journals and said they did not handle this correctly. And I think you have to be kicking against the establishment every now and then to because I think people like that. So, but I try to be very polite and objective and don't call people names. As you have more followers, I think you have to be very respectful for not, how do you call it? Like pushing down, or there's a term for it. That's not the correct term, but like if I would say, oh, this, here's the Twitter handle of an editor who incorrectly handled this, and it's a person with 200 followers, I feel that would be kicking down or pushing down. But I feel when a person has a Wikipedia page, they're famous enough to be sturdy enough to be able to be criticized publicly. But
Right.
when it's a paper and I just don't know these people, it might be a graduate student who was photoshopping, I'm not usually calling out which paper it is. I just show the image, and so I sort of still will protect the people who are on the paper. But occasionally when it's really, really bad, I will give a link to the paper, but I don't want my followers to harass particular people if they are on Twitter. So I keep an eye out on that. But you have to be very responsible. And, there's other more nasty people on Twitter who will send all their followers, all their fans to harass one person. And I feel that is just not how I want to operate. So I want to
Right.
be responsible, respectful, and also thank people. If people send me a tip, I will thank them. And I'm sure I make mistakes, but, I try to do my best.
Thank you so much for meeting with me and letting me hear more about your work. This is super exciting. So yeah.
You're very welcome. Yeah, no, it was my pleasure talking to you.